Hello. In the article shown above, the researchers focused on alanine scanning mutagenesis for analyzing protein functionality at single amino acid resolution and design of stabilized proteins. Standard site-directed mutagenesis is a challenging, expensive, and time-consuming process, considering that you're creating libraries of hundreds of mutants and there are PCR sequence errors like misannealing and tandem primer repeats during mutagenesis cloning. To improve this technique, they introduced a high-throughput mutagenesis pipeline that is a combined two-fragment PCR approach, where mutagenesis primers are used in two separate PCR reactions. So the standard mutagenesis technique is a one-fragment approach that relies on two primers in one PCR reaction, and this improved technique is a two-fragment approach where the two mutagenesis primers are separated into two PCR reactions and combined with one reverse primer. Then the two vector fragments generated in the separate PCRs are then combined in Gibson assembly. Based on their success with the arrestin-1 mutants, the high-throughput scanning mutagenesis was applied to two human GPCRs, CB2 and V2R. The amino acids of the target protein were changed to alanine, while alanines were exchanged for glycines for V2R or valines for CB2. They tested different DNA polymerases and optimized PCR conditions for 30 randomly chosen CB2 mutants, and they found out that the GC buffer illustrated all the expected fragments in all cases, and the HF buffer failed to multiply 4 fragments. They then performed 2 PCR reactions per mutant with half of the vector amplified in each reaction, and combined the 2 matching fragments that contained the 1 mutation. Bacteria were transformed with the resulting plasmid and plated, and the 1 clone per mutant was sent for sequencing. They successfully generated complete alanine scanning libraries of the 2 GPCRs. For V2R, 83% of the colonies sent for sequencing had the right sequences, and CB2 had 73% of the colonies that had the right sequences. This lower efficiency of CB2 was due to the failed sequencing and the primer misannealing, resulting in large deletions. They used the same two-fragment cloning approach for 2 plant membrane proteins, and they found out that 90% of the successfully sequenced DNA samples had the correct sequence. So in comparison to the standard method, the number of tandem repeats of the primer sequence before and after the mutation was drastically reduced, thus resulting in increased cloning efficiency. So 86% and 90% compared to 54% of successfully sequenced samples. The 2 separate PCR reactions amplified the 2 fragments of the target vector, and thus generated alanine scanning libraries of 400 single-point mutations in 6 weeks, with complete coverage of the protein sequence.